Hello, everyone. This is Dr. Mikky recording a lecture on Chapter 5, which is the Pharmacodynamics. So in this chapter, we are going to cover content on the what is the pharmacodynamics, and which focuses on which is the study and of the effect of the drug on the body, and goes into more details and in-depth covering on the dose response relationships and the interactions between the drug or medication and the receptors and the drug responses that do not involve receptors. There are some medications that may cause an effect or result in physiologic changes to the cells and tissues, and without having any interaction with any receptors. Also, we are going to cover the interpatient variability in drug responses, how patients respond differently to the same medication, or different people responds differently to same drug, which is called the interpatient variability in drug responses. And we will highlight part of what we discussed previous chapter, which is pharmacokinetics, about the therapeutic index. So what is pharmacodynamics? So it is how a medicine changes the body. In other words, it is the study of what drugs do to the body and how they do it. It will help us to predict if the drug will produce a change or response. It will ensure that the drug will provide safe and effective treatment. So a combination of drug guides and intuitive experience will guide safe treatment. Now, to accomplish the therapeutic objective, nurses must have an understanding, and this is a basic or Pharm Patho or Patho Pharm content for the undergrad. So at least at this level, we have to ascertain that we have a basic understanding of the pharmacodynamics. What is that pharmacodynamics? What are the components of this study, this type of study, and how the drug affects our body, and what are the inpatient variabilities in drug responses as we discuss the different medications thereafter. So the pharmacodynamics, so educating patients about their medications, making that the as needed or PRN decisions are made appropriately in a safe, effective way, evaluating patients for drug responses. We have to evaluate them for both the beneficial, the expected predicted outcome or positive outcome, or the positive responses from the drug, as well as the harmful effect. Collaborating with physicians and other practitioners like nurse practitioners and prescribers about the drug therapy. So examples of this would be like the vancomycin trough levels. The trough level should be drawn for vancomycin before or prior to the next scheduled dose. So the trough levels for vancomycin, it determines the dosage administration. Are we going to continue with the same dosage, or to hold it, increase or decrease the dosage? It's based on the vancomycin trough levels, and we have trough levels and medication or plasma levels for these meds. We have too many examples, and as we know, we will we discuss the medications either in this chapter or in the upcoming subsequent chapters. We will go over too many of these meds that requires either a trough level or plasma drug level, like the Dilantin, for example. We have to maintain it within range, which is called the therapeutic range. Here, it's different. it's called trough level. And we'll explain about these levels as we discuss concepts in this chapter of the pharmacodynamics. Another example would be like a PT-INR result for warfarin sodium Coumadin dosage. Coumadin dosage is determined, the dosage and frequency, or at least dosage, is determined based on the results of the PT-INR, and we focus on the INR more than the PT to maintain between 2 to 3 for the most part. And we explained about this concept of INR and warfarin on last week in chapter 4. The same thing with blood pressure parameters and the antihypertensive drugs, the dosage, the administration of the blood pressure medication to continue the same drug or to hold it or to decrease or increase the dosage of the blood pressure medication based on these parameters, which is blood pressure and heart rate. So if the patient's blood pressure is dropping or it's not within range and it's less than the normal or expected values then or parameters, then we have to hold the drug, for example. And vice versa in terms of that, if the blood pressure is still not under control and this dosage is not controlling the blood pressure, then we have to increase the dosage or the frequency. So the dose required to produce a therapeutic response can vary substantially among patients. That's why we have failure defined as the inter-patient variability. Now, measurements of inter-patient variability can be illustrated by examining the frequency distribution curve, which is on the right-hand side of the slide. And we'll go over this on the upcoming slides. Now, the frequency distribution curve, it's a graphical representation of number of patients responding to the drug action at different doses. So on the x-axis, you have the dosage of the medication, milligrams, and in the y-axis, you have the number of patients responding to the drug. Now, the peak of curve indicates the largest number of patients responding to that specific drug. But it doesn't show the magnitude of the response, but just give us the number of patients who responded to the drug at different dosages. So let's say that we collected the data for this inter-patient variation in drug responses and data from tests of hypothetical acid suppressant in like 100 patients. And the goal of the study is to determine the dosage required by each patient to elevate the graphic pH to, let's select to 5. So there will be a wide variability in dose or doses needed to produce the target response for the 100 subjects. So which is represented in the previous slide, hypothetically. So this is like hypothetical explanation of the effect of the drug and how many patients responded and what's the peak and what was the peak of which the peak of the curve indicates the largest number of patients responding to the drug. So the ED50, which is the dose that will produce a predefined intensity of response in 50% of the population. So that's the dose at the middle of the curve is termed the ED50, which is the dose that will produce a predefined intensity of response in 50% of the population. 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 50% of the So what are the clinical implications of intra-patient variability? So the initial dose of the drug is necessary or it's required, and it's like an approximation of what's expected or predicted effect. So the initial dose of drug is necessarily an approximation. So subsequent doses must be fine-tuned based on patient's response. So the ED50 is in a patient, we need to be increased or decreased after evaluating the patient response. So there is a relationship between the size of an administered dose and the intensity of the response produced. How does patient respond to varying doses of the drug? So we will probably have a simple answer for this one, saying that the antibiotic would kill more bacteria if the dose was increased from 10 milligrams to 20 milligrams. Another example would be the antihypertensive for high blood pressure medication. Antihypertensive medication would cause greater reduction in blood pressure if the dose was increased from 50 to 100 milligrams. So the dose response relationship, as we look at the dose response relationship, it determines the amount or the minimum amount of the drug we can use or prescribe. The maximum response of a drug can elicit and how much we need to increase the dosage to produce the desired increase in response. So again, the dose response relationship determines the minimum amount of drug we can use or prescribe, the maximum response a drug can elicit, and how much we need to increase the dosage to produce the desired increase in response. So as the dosage increases, the response becomes progressively larger. So the dose response relationship is graded as an increase in dose that will result in increase in response. So there are three distinctive phases of a drug response curve that indicate the essential pharmacodynamic principles. So the three phases of a drug response curve that indicate essential pharmacodynamic principles. Phase one, the flat portion of the curve. Few target cells have been affected, small doses will not produce a therapeutic effect. At phase two, straight line portion of the curve, linear relationship. So if the dose is doubled, then twice as much response is obtained. So you give a 50 milligrams of antihypertensive drug, if you double it to 100 milligrams, then you expect the double also change of the response. In stage 3, that's when the plateau is reached, and increasing the dose will not increase the intensity of the response. The response will be maintained at the same level. So no matter how much you increase the dosage, the response or resultant response will be maintained at the same level. So in phase 1, the curve is relatively flat. And doses are too low to elicit a significant response. At phase 2, the curve climbs and upwards as bigger doses elicit a corresponding increase in response. And then in phase 3, and then the curve level is off, and bigger doses are unable to elicit a further increase in response. So in phase 1, it's not indicated in A because very low doses cannot be shown on a linear scale. So this is a what is called graded dose response relationship. So this is a graphical visualization of the differences in responses to medications in a single patient, obtained by observing and measuring patients' response at different doses of the drug. So in phase 3, we said that while the increase in dose size will not elicit any change in the intensity of the drug response, and it's because the plateau has reached. So the plateau has reached or is reached because all target receptors for the drug are or became occupied, filled up with the medication, or interacted already with the medication. So all target receptors for the drug are occupied, completely saturated, in other words. So the drug has brought 100% relief, which means that more Excedrin, for example, will not produce more relief if the migraine was completely relieved. So if the required dosage is, let's say, 600 of ibuprofen milligrams tablet, PO, every 6 hours, if taking the 600 milligrams PO tablet relieves patient's pain, so an increase in dosage is not required. And in other words, that because the plateau has reached, so because that dosage was enough and sufficient to to relieve patients' pain level. So again, the drug has 100% relief, so which means that the drug or extra or additional amount or dosage or increase the drug dosage will not elicit any further change or in the response because the plateau has or is reached. Now, the in this phase, the nurse should be aware that there is no additional therapeutic effect, but increasing the dose at this point may produce more side effects. Plateau has reached, effective has been obtained, or effectiveness has been obtained of the drug response, any further increase of the drug dosage at this point will not affect the response because we already have really, reached and obtained the response or desired response, but it will produce more side effects. Therapeutic index. So let's just look at the ED50. What is ED50? Lethal dose and effective dose. Effective dose D50 and lethal dose D50, ED50. So the median effective dose, ED50, is the middle of frequency distribution curve, the middle of the curve, the highest point of the curve in the middle. Dose which is the dose, this is a median effective dose, ED50. It's the dose that produces therapeutic response in 50% of a population group of patients. Sometimes we call that average, sometimes the standard dose or average dose. And many patients, they may require more or less of that dosage. So the nurses, in terms of the nursing skill, which is critical in determining if a average dose is effective by doing the patient observation, taking the vital signs, monitoring the lab data and lab values. Now, what is the ED50, the lethal dose, LD50? The lethal dose, the median lethal dose, LD50, used to assess safety of the drug, shown on frequency distribution curve, and it determined in preclinical trials. During the preclinical trials, we'll cover this in Patho Pharm 2. So it is the lethal dose in 50% of group of animals. This is done usually on preclinical trials, on non-human subjects, and cannot be experimentally determined in humans. The median toxicity dose, which is the TD50, median toxicity dose, and which is a dose that will produce a given toxicity in 50% of groups of patients. So because the LD50 lethal dose cannot be experimentally determined in humans, so the median toxicity dose, the TD50, is a much more practical value for a clinical practice. The TD50 toxicity dose value may be extrapolated from the animal data or based on adverse effects recorded in patient clinical trials. So therapeutic dose, index, TI, which is the ratio of the LD50, which is the average lethal dose, to 50% of the animals treated, to the ED50, which is the dose of drugs that will kill 50% of group of animals. In the lab during the preclinical phase of the drug development process. So the TI, which is the relationship between the lethal dose and the effective dose, the ED50. So the TI equals LD50 divided by ED50. And that's how we calculate the therapeutic dose. Now, there are characteristics of uh, um, uh, two characteristics or properties of the drug that are re that are revealed in dose response curves. Usually, we see an drug response curve like potency versus um, efficacy. So, drug is potent, but is it what is it, the drug's efficacy? So these two are concepts or characteristics are two important properties of the drug when we discuss or go over the drug response curves or relationship. So there are two concepts that will describe the drug's activity. For example, some antibiotics or antihypertensive drugs lower the blood pressure to a greater extent, more effective level or extent than others. Antineoplastic agents like anti-cancer drugs, some of these agents or medications, they will kill more cancer cells than others. Analgesics in the same class, they may have different characteristics of potency and efficacy. So so for example, the drugs in the same class are effective at different doses. One antibiotic may be effective at a dose of 1 milligram per kilogram, whereas another drug is more effective at 100 milligrams per kilogram, even though these medications belong to the same classification or the same class. So, so this graph shows the difference between the potency and efficacy. Efficacy, which is the maximal effect or efficacy, which is an index of the maximal response the drug can produce. Efficacy of the drug is indicated by the height of its dose response curve. So if you have this example, for example, this example is that the pyridine, for example, we have here under the A, is that it has a greater efficacy than pentazocine. Efficacy is an important quality in a drug. Efficacy is a very important quality for any drug. The potency, which is the index of how much or how much a drug must be administered to result in or elicit the desired response. And in this example, under B, you'll find that achieving the pain relief with the methylpyridine, like pethidine, Demerol, or methylpyridine, this is the same drug. Achieving pain relief with this drug, methylpyridine, requires higher doses than with morphine. So to reach the same potency level, if the patient takes methylpyridine, or I'm sorry, morphine, then you need too many doses of that, or large amount of this dose, methylpyridine, to reach the same potency level of morphine. So here we can say that morphine is more potent than methylpyridine, because the potency is the index of how much the drug must be administered to elicit the desired response. So potency is not an important quality in a drug. So if we say that morphine is more potent than mifepristone, so we will note here is that if administered in sufficiently high doses, mifepristone can produce just as much pain relief as morphine. So that's why the most important quality here is the efficacy. Efficacy is such is very important quality in a drug. The potency is usually not an important quality in a drug. So again, just recap here that the potency is the amount of drug you must give to elicit a response or effect. So drugs with high potency produce a therapeutic effect at a lower dose compared to another drug in the same class. So we can say morphine is more potent than mifepristone because the drug can produce a higher therapeutic or can produce its therapeutic effect at a lower dose compared to mifepristone. The calcium channel blockers, which is another drug that will control the blood pressure for patients with high blood pressure, CCB, calcium channel blockers like amlodipine, Norvasc. It produces its effect or therapeutic effect at 10 milligrams than another drug like nifedipine, which is Procardia, which is from the same class. It is both of both of these meds, amlodipine and nifedipine, they belong to the same drug class called calcium channel blockers. But you can control blood pressures with amlodipine at 10 milligrams dosage, but nifedipine, the dosage should be at least 180 milligrams. So amlodipine is more potent than nifedipine. Again, this concept or the quality of the drug, not that much of importance compared to the efficacy. So because there are reasons why we have to prescribe amlodipine to the A patient, but the another patient or B patient supposed to be on nifedipine. So there are always a scientific reason why different medications from the same class are prescribed to different patients. So again, the potency as a quality of the drug is not important as the efficacy. Efficacy is very important, which is the efficacy, which is the magnitude or maximal response that can be produced from a particular drug. So maximal efficacy, which is the largest or greatest maximal effect or response that the drug can produce, which is always the highest point on the curve, which is in figure A, previous slide. So you measure the intensity of response what patients need. And very high maximal efficacy is not always more desirable. Now, for example, aspirin versus morphine for different pain levels. Another example, you compare like Lasix versus the hydrochlorothiazide for different diuretic purposes. So we'll cover more of these examples and specific drugs in the subsequent upcoming chapters. So another recap on this too is that potency is rarely, we said potency is rarely an important characteristic of the drug. So it is rare for a specific drug or drug to be so lacking in potency that doses of inconvenient magnitude should be given. In other words, that there is this potency quality of the drug is not as important as the efficacy, but both completely are independent. So So when we say efficacy and potency, they are completely independent qualities of the drug. Ibuprofen, for example, ibuprofen 200 milligrams versus aspirin 650 milligrams. We can see ibuprofen is more potent than aspirin, but both release pain at a certain doses, at a certain dose, both equally effective. So, however, for severe pain, neither of the above two medications of the ibuprofen or the aspirin has sufficient efficacy in relieving severe pain. So that's why we will move and go and prescribe morphine. So morphine has greater efficacy than aspirin and ibuprofen. So the efficacy is more important than potency. Dose or drug receptor interactions. Receptors So the receptor theory predicts that the response of the drug is proportional to the concentration of receptors that are bound or occupied by the drug. So technically, receptors can induce enzymes, ribosomes, and others. So the term receptor is generally reserved for the body's own receptors to bind endogenous molecules like hormones, neurotransmitters, growth factors, and other molecules. And most drug receptors are protein agonists. Now, binding of a drug to its receptor is usually irreversible, and the drug, it can stimulate or inhibit normal activity. So when the drug binds to a receptor, it will either mimic or block it. So the action of the endogenous regulatory molecules and increase or decrease the rate of physiologic activity normally controlled by that receptor. And this is one of the most important concepts in drug receptor interactions, is that when a drug binds to a receptor, it will either mimic or block that receptor. So the drug may trigger second messenger events, like activation of specific G proteins and associated enzymes. It may initiate the drug action. And below here is that, an example of the interaction of drug receptors for norepinephrine. So this is the norepi, and we have two different chemicals. One, natural, intrinsic, endogenous, comes for the nerves, the norepinephrine released from our nerves, from our body. And the drug synthetically is manufactured and synthetically given to the patient, norepinephrine, and is given to the patient or administered to the patient. So you can tell from both um, uh, the structure of both medications, they're identical in terms of that both of them can bind to the same receptor, which is the R. So this is an epinephrine receptor, and if that's the intrinsic, natural, or if it was a drug, both will easily can bind to that, to this receptor. So under physiologic conditions, the cardiac output can be increased by the binding of epinephrine, norepinephrine to receptors on the heart. So in this case, we can say that these same receptors can be acted by the drugs. So in which that this drug will mimic the actions of norepinephrine. And then eventually it will result in the same effect as if it was synthetic. I mean, it was in a natural intrinsic one. So when we administer the norepinephrine, we anticipate that it's going to result in the same physiologic activity or effect, which is to increase cardiac output. Blocking the action will result in reduction in cardiac output. So activation of the response by the norepinephrine is going to result in increase in cardiac output, and blocking that action or emit like a different another medication that will block the norepi receptor, this specific receptor, then it will result in a reduction in cardiac output. But the drug, for this specific drug, norepinephrine, synthesized and manufactured to be administered, either it's usually IV, it will be given and it will result in increase in cardiac output because it has the same expected physiologic effect as if it was the endogenous intrinsic one. 4 primary receptor families. So just go over the 4 receptor families that we have: the cell membrane embedded enzymes, like, for example, insulin; ligand-gated ion channels, like acetylcholine; G-protein coupled receptor systems, like norepinephrine, serotonin, histamine; and the last one is the transcription factors, like thyroid hormones, steroid hormones. So receptors are normal points of control of physiologic processes. Their normal function is to bind endogenous molecules such as hormones, neurotransmitters, and growth hormones. And under physiologic conditions, receptor function is regulated by molecules supplied by the body. So the drugs can only mimic or block the body's own regulatory molecule. The drugs cannot give cells new functions. It's either mimic or block these molecules. So drugs produce their therapeutic effects by helping the body use its pre-existing capabilities, which is like receptors, targets. So in theory, it should be possible to synthesize drug that can alter the rate of any biologic process for which receptors exist, which is the same concept that we have discussed earlier about the norepinephrine and how's the manufactured synthesized drug called norepinephrine was manufactured based on the qualities and the exact structure of the norepinephrine that's being produced endogenously, so to result in the same effect, which is increased blood cardiac output. So the more selective the drug is, the fewer side effects it will produce. And this is also concept has been discussed in during the first week class. So we have different receptors. We have like alpha 1, alpha 2, beta 1, and beta 2 receptors. So receptors make selectivity possible. Each type of receptor participates in the regulation of just a few processes. So it's based on what is called the lock and key mechanism. Each receptor binds to specific chemicals or specific drugs, medications. So the receptors, so our body has receptors for each one of the neurotransmitters, each of the hormones, all other molecules the body uses to regulate physiologic processes. It has its own receptor, very specific to that, chemical or to that neurotransmitter or to the hormone or any other medication or molecule. The receptor is only designed to accept binding to that specific molecule. Simple occupancy theory, modified occupancy theory, so affinity and intrinsic activity. So the affinity, which is the strength of the attraction of that specific molecule or drug to the receptors. Drug's affinity is reflected in its potency. Drugs with high affinity are very potent drugs because they bind to receptors at low concentration. However, intrinsic activity is the ability of the drug to activate a receptor upon binding. So when the drug binds to a receptor, then the ability of that drug to activate the receptor, that is intrinsic activity. The drug can cause, or drugs that cause the intense receptor activation result in intense responses. So drugs that cause the intense receptor activation, it will result in intense responses. So intense activation will result in intense responses. So the drug's intrinsic activity is reflected in its maximal efficacy. Agonists or the ags are molecules that activate receptors. They will bind to a receptor, a drug that activate a receptor and produce the same type of response as the endogenous substance. And sometimes it will produce greater or heightened maximal response. Like, for example, the ethanol, it binds to the acetylcholine receptor in the ENS autonomic nervous system, and it will produce same actions as acetylcholine. The endogenous regulators are considered agonists. The endogenous, intrinsic regulators are considered agonists. The agonists have both affinity and high intrinsic activity, like dobutamine, for example. Dobutamine, it's a synthetic or synthesized, it's a drug medication, heavily used or commonly used in the critical care setting. Dobutamine mimics the norepinephrine at cardiac receptors. So norepinephrine, as an intrinsic endogenous chemical, the dobutamine synthetic drug, it mimics that norepinephrine at the same receptors, the cardiac receptors. And the agonist doesn't mean that the agonist is always going to be to make the processes go faster. Sometimes it will make it go slower. Like, for example, acetylcholine slows the heart rate. So there are also there is a partial agonist, but we are not going to cover that in this. It's not within the scope of this class to go over the um, uh, advanced patho or patho so there's something called like a partial agonist, which will bind the receptor, and these are the agonists that have only moderate intrinsic activity. So we'll move on to the antagonists. The antagonists, they produce their effects by preventing receptor activation by endogenous regulatory molecules and drugs. So it's a drug that will occupy a receptor and prevent the endogenous chemical from binding to produce its action. An example of this one would be like atropine. It will compete with the acetylcholine for certain receptors in the autonomic nervous system. Large atropine doses may block the effect of acetylcholine because acetylcholine cannot reach its receptor sites. Now, antagonists often compete with agonists for the receptor binding sites. No effect of their own on receptor function. So the antagonists, again, they have no effects of their own on receptor function. Antagonists have affinity but no intrinsic activity. They do not cause receptor activation but will result in pharmacologic effects by preventing the activation of receptors by agonists. So if there is no agonist present, an antagonist will have no observable effect. So no observable effect from the antagonist if there is no agonist present. We have functional antagonists, which will inhibit the effect of an agonist not by competing for a receptor, but by changing pharmacokinetic factors. We call that functional antagonist. These antagonists that will inhibit the effect of an agonist not by competing for a receptor, but by changing the pharmacokinetics or pharmacokinetic factors. So not all antagonists are associated with receptors. Functional antagonists inhibit the effect of an agonist, not competing for a receptor, but changing the pharmacokinetic factors, such as the antagonist may slow the absorption of an agonist. Antagonists may change the pH and neutralize the agonist, or speeding up the metabolism or excretion. So the resultant effect of this antagonist as a functional antagonist, not competing with a receptor, but changing the absorption, slow the absorption of an agonist, or change the pH, or neutralizing the agonist, or speeding or accelerating the metabolism or excretion of the agonist. Also, there is what is called non-competitive antagonist, competitive antagonist, and we are not going to cover that in this class. So the regulation of receptor sensitivity, the receptors are dynamic cell components. So if the receptors are dynamic cell components, so the number of receptors on cell surface and sensitivity to agonists can change in response to continuous activation or continuous inhibition. And sometimes the continuous exposure to agonists will result in either desensitized less responsive, or sometimes down regulation. And these concepts may seem complicated, but we cover this concept in the hormonal or the chapter which includes all the hormones. And the continuous exposure to an antagonist may result in hypersensitive due to the synthesis of more receptors. So drug responses that do not involve receptors, we have examples of this, of the receptorless drugs, like antacids, in which that they have a direct interaction with the stomach acid. Antiseptics, they have um, um, result in precipitating bacterial proteins. Or its effect on bacterial proteins. Saline laxative. These laxatives will retain water in the small intestine. Chelating agent. Chelating agents, these are the medications that are going to bind to um, specific molecules and get rid of them. And most of the time, it's usually chelating agents in the GI tract, or sometimes even in the blood. So chelating agents, these are they form what is called complexes with toxic molecules, and which will be released in the with bowel movement or in urine, or most of the time either bowel or urine. So these chelating agents, they help the body to get rid of these toxic materials.